Clemson scientist Cheryl Ingram-Smith has been awarded a $424,000 NIH grant to study the inner workings of a parasite called Entamoeba histolytica, which infects 500 million people annually around the world and causes up to 100,000 deaths. Ingram-Smith's research is focusing on a pair of unusual enzymes that appear to enable the parasite to thrive.
Entamoeba is unusual in, first of all, it's an amoeba, so it just kind of oozes around. It's just fun to watch under the microscope. Most eukaryotes, including both plants and other cells, have what's called a mitochondria. And that's basically a powerhouse. And so the mitochondria is where you make ATP. And ATP it's just basically the money of the cell. It's energy money. And so most processes that go on in the cell use ATP for energy. So the mitochondria is a powerhouse that just deals out ATP. In normal cells, you take glucose, you break it down by a process called glycolysis, and that breaks glucose down partway, and you get just a little bit of ATP out of that. Then you go to the mitochondria, and then the mitochondria breaks it down the rest of the way and makes a lot of ATP. Entamoeba can't do that. Entamoeba doesn't have a mitochondria at all. So it takes glucose, goes through this process, glycolysis, and gets out to ATP. And so it not only can't make a lot of stuff that it should be able to make, but it can't break down glucose well either. So I'm studying a couple enzymes that we think help glycolysis work better.